Okay, I'm going to set the scene with an overview of how DSI is changing how we conserve and use genetic resources. Just to set the context, so you know where we're coming from, the core mission of CGIAR is to help developing countries, largely by delivering the 4 so-called non-monetary benefits referred to in the treaty. We work in partnership with them all the way through the value chain, from identifying what their problems are, all the way through to developing and scaling up the products. Now, DSI have become essential for effective conservation and sustainable use of PGRFA. So to help developing countries better, we have to use DSI, and we have to help developing countries use DSI. The question for which we don't really have an answer is, how do we get the monetary benefit sharing right? Our primary concern here is that any monetary benefit sharing system must not interfere with the creation and sharing of non-monetary benefits, which are economically far greater. So, based on the experience we've had with DSI, we have identified 3 features that are real game changers in terms of benefit sharing. The first is that DSI delinks the use of genetic resources for research from the use of genetic resources for development of products. The second is that DSI-related technologies enable the in vitro and in silico replication of units of heredity. And third is that DSI enables discovery of unrelated genetic resources containing the same sequences. I'm going to go through each of these briefly, one by one, before the next presentations go into more detail. So first, the point here is that genetic resources are now used much more for upstream research. That research creates the knowledge and the tools that we need for more effective and efficient development of products. So that research creates huge benefits, even though the genetic resources used are almost never incorporated into the final products. The upstream research has become a much more significant core component of the development pathway. And I put in here as the first example Arabidopsis thaliana, which is a weed primarily of developed countries. Essentially, all our basic knowledge of the molecular biology of plants comes from research on this weed species. I think it's fair to say that without this research we would not be having this discussion about DSI and ABS, because we would have no system of biotech to improve crops. Next example is the use of reference genomes. The first monocot reference genome was Nippon Bari, a Japanese temperate rice variety. Sequencing began in 1997 in a project involving 10 countries, and it was 5 years before the first draft sequence was published. And even now, they're still trying to improve it. So investment in upstream research on Nippon Bari has created massive benefits for rice breeding, although the Nippon Bari genome itself is not incorporated into any tropical rice products. Thirdly, we have genome-wide association studies, where you take hundreds or preferably thousands of samples from different countries. You sequence or at least genotype them all, and you evaluate them phenotypically. By looking for statistical associations between genetic and phenotypic variants, you can begin to identify the genes that control the trait you're looking at. This is a great technology for CGIAR and developing countries, because if we in CGIAR genotype or sequence all these samples and make the data available to developing countries, then with very little extra training, they can conduct their own phenotypic studies in their own countries and begin to understand the genetic control of their own traits that are important to them in the environments that are important to them. Again, most of these genetic resources won't be incorporated into products, but the knowledge gained through the research transforms their ability to develop products. Our message to the CBD was that it's a mistake to tie benefit sharing just to the few genetic resources that are incorporated into products through bilateral ABS. That's not a problem for the treaty, because its MLS already captures the value created by the upstream research when it is realized through the commercialization of PGRF8 products. So you're already a step ahead of the CBD. But the MLS does not capture monetary benefits when the knowledge and tools are used to develop products derived only from PGRFA that are not in the MLS. Nor does it capture monetary benefits from commercialization of non-PGRFA products, such as information and tools. Next, we come to a point that I've been told is a little bit philosophical, so it's appropriate that one of the key recent papers on this has been published in the Philosophical Transactions of the Royal Society. This paper was published in 2016, but as far as I know, the concept that DNA can be viewed as information is at least 50 years old. When you think about it, the DNA in offspring is just a copy of the DNA in the parent. The atoms are different, the molecules are different, the actual material is different. The only thing that's the same, the only thing that's transmitted across generations is the sequence, which is information rather than material. When we talk about products that incorporate genetic material from the MLS, we don't mean they incorporate the actual material DNA, we just mean they incorporate copies of the DNA with the same sequence, that is, with the same information content. So you can view the unit of heredity as information, and DNA is just the material that carries the information. And here is why that's important. Conventionally, we think of ABS as being based on genetic material incorporated into products through in vivo replication of DNA by mitosis and meiosis. But with genetic modification and gene editing, we can replicate DNA in vitro. Here, DNA is still the carrier of information, but we can replicate it without mitosis and meiosis. With synthetic biology, we can go a step further away, replicating DNA sequences in databases before reverting to material products. So here, computer disks, not DNA, are the material carrier of information. Now the CBD and the treaty are both silent on how the information content of units of heredity is stored or replicated. So it seems reasonable to view all of these as being within scope, although that does not appear to be how the SMTA is worded. But once the information is on a computer disk, it becomes effectively impossible to police or trace or monitor. So an ABS system that relies on the incorporation of units of heredity into products becomes unworkable. Finally, we come to the problem that DSI enables discovery of unrelated genetic resources containing the same sequences. I think this has been recognized for several years as a loophole in the SMTA. You can get material under an SMTA, use it to discover and sequence a valuable gene, and then find the same sequence in different material that is not subject to an SMTA. This obviously creates a strong temptation to bypass the SMTA's provision by doing the research on MLS material, but doing the product development on non-MLS material. For example, basmati rice is really important to India and Pakistan, and Thai jasmine rice is really important to Thailand. They actually have identical sequences of the major aroma gene, and you can find identical sequences also in aromatic rice from other countries, such as Nepal and Bhutan. In its normal state, this gene produces an enzyme that breaks down the major aromatic compound so that its concentration remains low. If you disable that gene, the concentration of the aromatic compound builds up, so you get an aromatic rice. So essentially any alteration to the gene that stops it working results in aromatic rice. So you can get similar sequences with identical effect in many other countries. I've listed here just a few, and you can invent many other sequences that stop the gene working as well. So I'll finish off this introductory presentation with this take-home message. Monetary ABS can be fair and equitable only in a multilateral system that delinks payments from incorporation into products, for the three reasons I've gone through. Value creation relies on the use of diverse genetic resources to build knowledge and tools. In vitro and in silico replication of units of heredity can make it impossible to police incorporation into products. And multiple origins of the same sequence incentivizes users to bypass ABS. Thank you. Now let's pass to the next presenter.